Our next lecture is from Tom Rando, who's the Deputy Director for the Stanford Center on Longevity.
Great. Again, thank you to the organizer for inviting me. It's a great pleasure to be here for the whole workshop, and then especially to Ava and Andrea for today's workshop on longevity medicine. We do a lot of basic work in my lab, but we do some work that I consider more translational on how to get the basic biology into the aspect of health span. And I thought today I'd talk about exercise, which is something that we've been very interested in for some time, and how that translates into, great, thank you, into, is that a pointer? Nope. Sorry. Into better health. And so a lot of what I'll talk about today is, not surprisingly, will be inflammation, and how with exercise there are reversions to kind of more youthful states in terms of inflammation. Some talk about myokines secreted by muscle. I think it'll be interesting, but some maybe potentially interesting players in the mix. Here we go. So we work primarily on skeletal muscle, and we're interested in muscle, as I'll talk about, in terms of its role in general health, but we're also interested as a model for tissue repair and regeneration, because it is a largely stable tissue, but it has remarkable regenerative potential, and that's illustrated here. So in the top panel is a cross-section of a normal skeletal muscle. Each of those polygonal structures is so-called muscle fiber, and they're multinucleated large cells that are responsible for your movement. But within that mix, there are also, I'll show this, very rare muscle stem cells that are responsible for this regenerative potential. So below that, you see a totally necrotic tissue after a chemical injury, and then about a week later, you can see the formation of these small cells called myotubes, which are en route to forming the larger myofibers. And so this is a really dramatic response that can occur in this muscle in response to injury. And the cell responsible for that is shown here with this arrow, which is this one tiny little stem cell, or so-called satellite cell, that sits in the muscle in a quiescent state. It just is there kind of as a reserve cell, but it's responsible when the muscle is injured to undergo this massive proliferation, differentiation, and make new muscle. Now, that's something that happens very well when we're young, but as we get older, there are changes in the ability of our tissues to repair. And we know this quite well across all tissues, that with time, with age, tissue repair is less efficient and less effective, and often with some excessive scar formation. And so this is just an example in the mouse, a young mouse and an old mouse. Again, during this intermediate stage of regeneration, when there are these myogenic cells shown here, sort of in purple, but then with age, I can't see the screen, so you have to take this look for me. To the right of that is an older muscle where you see, again, fewer and smaller of these regenerating muscle cells and more of this interstitial connective tissue, which is in green. And that's what ultimately will lead to more fibrosis in the tissue. And you see this in tissues across the body, whether it's bone or skin or muscle with age. And this is just illustrated here, again, showing that with age, if you injure and regenerate a muscle, a young muscle will regenerate very well, whereas with age, an older muscle will regenerate, but there'll be more scarring. And so you end up with a less effective tissue and a stiffer tissue. So, a question we've posed since we've been studying the biology of this process and this aging is it possible to restore youthful properties to old tissues, to basically get aged muscle to regenerate as well as young muscle? And we were inspired early on in my career. I was close friends with a gentleman, John Faulkner, who's unfortunately passed away, but he was a real kind of mentor to me in terms of muscle biology. He did this fairly simple experiment of transplanting young muscle or old muscle into a young host or an old host. And what was important was the age of the donor, the muscle that was transplanted, was less important in terms of how effective it was in restoring new muscle as the age of the host. So what you see on the left is this is a young host on the left that has received either a young muscle transplant or an old muscle transplant, and you see very effective regeneration. Whereas the old host on the right, regardless of the age of the donor, you see this kind of fibrotic regeneration. So this led us, this is in the early 2000s, to pursue these studies of parabiosis, or so-called heterochronic parabiosis, asking these questions about what the environment in which a tissue repairs, how effective, how important is that for determining how good the tissue repairs. Basically, we did these connections, and so you get, in the middle of these heterochronic pairs, you get the old mouse exposed to the circulating environment of the young mouse, and the young mouse exposed to the circulating environment of the old mouse. And the question always was, what effect does that milieu they have on the regeneration of the tissue. So, and what we found impressively early was the old mouse exposed to the circulating environment of the young mouse showed regeneration that was as good as what we see in a young mouse. So very little fibrosis, very good myogenesis, and it really kind of looked like it rejuvenated the tissue. So we showed this initially in muscle, we looked in liver. A couple of years later, we looked at this in brain with our colleague Tony Weiss-Corey, and then over the last decade or so, people have looked at this in a variety of different tissues and shown this kind of rejuvenation of tissue function and tissue repair through this process of heterochronic parabiosis. Okay, so as we're doing this, we're obviously thinking, this is not a very translatable experimental paradigm in terms of parabiosing people. Are there interventions that can restore youthful regenerative potential to individuals, whether it's an animal or a human? And we've studied kind of the two probably most well-studied and popular interventions, which are diet and exercise. But I'll talk today about work that we've done with exercise. So this is an article from the New York Times from a few years ago. So I'm a runner, so I like the mathematics here, although I don't think he has the mathematics exactly right. I don't think you can just keep adding years of life by adding years of running, but it's promising to think that. But what we do know, of course, is that running and that's kind of aerobic exercise has a huge impact on health span, a huge impact on the prevention of age-related diseases through physical activity. What's really we don't know is kind of what the extent of that is, what the mediators are. These are really important. I think for many people, what they really want us to do is to find the drug they can take to get the benefit of exercise without having to run. So I hope that never happens, but it may happen. We're heading in that direction. And so our question really was, does exercise, this kind of physical activity, does it enhance the regenerative potential of a tissue, in this case muscle? So we've used these wheels, just voluntary wheel running, which mice will get on and run, and they'll run for miles a night, really extensive running, young and old mice, and we've asked what effect does that kind of several weeks of voluntary wheel running have on the regenerative potential and on the functionality of these stem cells in the tissue. So we found, this was one of my favorite experimental reports ever published. So we do these studies with wheel running, and we get a lot of criticism from reviewers, from exercise physiologists, who might be right saying that this is not really exercise, because mice don't really think of exercise, and they do this because they're in a cage, and it's some kind of stereotypical weird behavior because they're caged. Well, this group from the Netherlands did this perfect experiment. They took a wheel, they put it in their backyard, they put a webcam on it, and animals came out at night and ran on the wheel. So animals like to run, and they would have rats and rabbits, and they had a frog, and so this is something this animals seem to like to do. Anyway, so we use this as a paradigm for exercise, and this is work from a MD PhD student, Jamie Brett, and a postdoc, Marina Arjona, who in fact showed that just 3 weeks of voluntary wheel running has a huge impact on regenerative potential of muscle. And so the measure here is kind of a regenerative index. How good is the muscle? How little is the fibrosis? And you see on the left, you see with young, exercise has little effect. But with age, there's a marked decrease in the regenerative potential, and the exercise has a marked effect on the improvement of that regeneration. So Jamie and Marina, they looked at the stem cells initially, just looking at the transcriptome, and what they found is the gene in these stem cells that was most highly upregulated in response to exercise and declined with age was cyclin D1. Now this was first a real surprise because these cells are not cycling, and cyclin D1 is clearly playing a key role in the cell cycle, and these cells are quiescent if there's no injury. So that was a surprise, but it turns out cyclin D1 has a lot of non-canonical roles, one of which is as a transcriptional co-repressor. So it represses genes. So we asked the question, if we look at the genes that are repressed in young and then go up with age as cyclin D1 goes down, or that are repressed in response to exercise when cyclin D1 goes up, or are increased when we knock out cyclin D1 in these cells, we basically looked and asked, what is the signature of the transcription factors that kind of encompass those genes? And it turned out to be very few transcription factors, actually two big transcription factors, TGF-beta through SMAD3 and NF-kappa B through REL. So we focused on TGF-beta because we had additional data saying that that would be important. And so this is just looking at transcription factor targets. If you look on the left, TGF-beta signaling was the most anti-correlated with cyclin D1, and its transcriptional regulator, SMAD3, was again the most transcriptionally anti-correlated with cyclin D1. So it really seemed as if cyclin D1 was suppressing this TGF-beta pathway and that with age, as cyclin D1 goes down, TGF-beta signaling goes up. So we asked if that were, we tested that hypothesis by using pharmacologic inhibitors of TGF-beta pathway, this LY drug. And here we're looking at a measure of stem cell function. The dotted line shows the youthful level. It goes down with age, but if we inhibit TGF-beta, that youthful signature comes back up. So we see really a restoration of youthful stem cell function by inhibiting this one pathway. If we take animals in which we've knocked down cyclin D1 just in muscle stem cells, again, inhibiting TGF-beta signaling restored those cells to a wild-type level. So it really seemed to be acting downstream of cyclin D1. And then finally, we created a mouse in which we could try to reverse the effect of aging, which is the downregulation of cyclin D1, by making a mouse in which we could inducibly turn on cyclin D1 just in muscle stem cells at any time we wanted. So we made the mice, we aged them for 2 years, we turned on cyclin D1 in these cells for just a week, and again, we looked at the stem cell function, and even just 1 week of induction of this one gene led to a marked improvement in the function of these aged stem cells. So this is our model in which we have young animals with high levels of cyclin D1 in these cells, suppressing TGF-beta signaling. With age, cyclin D1 goes down, TGF-beta signaling goes up. That inhibits muscle regeneration, it promotes fibrosis. And one of the things that exercise does is it restores cyclin D1 levels to these aged stem cells and restores muscle regeneration. So that's kind of the model that we've been working on. But one interesting finding from that paper that's actually in the supplement, but I think it's an important finding, is that we could take old mice that we've exercised, take their blood, transfuse it into an old mice that has not been exercised, and see the benefits of exercise. So this really says there's circulating factors in the blood. It's not just the local effect of the activity, but actually producing factors that are going into the blood that are having this beneficial effect. And then a couple of years later, with our colleague Tony Weiss-Corey, we looked at the same thing in the brain. If we look at exercise plasma from an exercised mouse, transfuse it into a non-exercised animal, we can see an increase in neurogenesis, an increase in neurostem cell activity from the circulating plasma. So what I want to finish with is just talking about some work we've done recently, asking in a little more detail, what are the mediators? Can we begin to understand what the cell-cell communication is, what's being secreted by muscle, and how the cells are responding to exercise in stem cells throughout the body. So this was a fairly massive experiment, and this was done in collaboration with my colleague Anne Brunet at Stanford, who works on neural stem cells, and my colleague Peggy Goodell at Baylor, who works on hematopoietic stem cells. So we took a cohort of mice, young and old, with and without exercise. The ones that exercised did so for 5 weeks, and at the end of that time, we took cells within the stem cells and the niche cells from 3 compartments: the brain, the blood, and the muscle. But then we also took blood cells and looked at and did single-cell RNA-seq on all those cells. We took white blood cells, did single-cell RNA-seq on those. We took plasma, we did proteomics on that. And I'll mention at the end, we took myofibers, and I'll come back to that, and did RNA-seq. But I'll start just by talking about the stem cell findings that we had. So this is a UMAP just showing all the single cells. We sequenced more than 400,000 cells, and just showing them in this UMAP distributed that are mostly segregated by tissue. Even cells that are similar from one tissue to the next, like endothelial cells or smooth muscle cells, they segregated by tissues, except for the immune cells. And a lot of the story that emerged from this study is the role of immune cells and inflammation in aging and in response to exercise. So in particular, we found that monocytes and macrophages are a prominent signature of tissue aging in these 3 stem cell compartments and respond, in fact, to exercise. So we looked at the monocytes and macrophages, we looked at all the cells, and we said, which genes are changed with age and are then rejuvenated with exercise? So if they go up with age, they go down with exercise, or vice versa. And this is just a kind of a rank order of that. And one interesting thing is that blue is the cells from muscle. There are a lot of muscle cells that show this dramatic response to exercise. But the most prominent, the one that is the most affected, are these monocytes. So tissue-resident monocytes, they have dramatic changes in their gene expression with age, and the immune cells, respond to exercise most prominently. So if we look at this inflammatory signature on top and just look at pathway analysis of these different pathways, again, what we're looking at here are genes that change with age and respond to exercise. And again, the monocytes in the muscle uniformly respond to exercise in terms of their kind of rejuvenating features. And just to note on the right, it's also true of monocytes in the blood. So really to start drawing our attention to this population of monocytes and macrophages, so using this chord plot, using cell chat, we can sort of predict what the monocytes and macrophages, what they're signaling to, which cells they're signaling to, and which ligands and receptors they're using, and conversely, what ligands and receptors they're receiving from cells in the muscle. And basically, there's a lot of information here, but on the left is the young control, and these are ligands that are either expressed on macrophages and monocytes, or are expressed on the other cells that signal to monocytes and macrophages. The middle panel shows what that looks like with age. And there's just a dramatic simplification of this signaling between macrophages and monocytes and other cells in the tissue. And then on the right is what happens to those cells in response to exercise. I mean, they really restore this much more complex, much more detailed cell-cell signaling. So, we're really interested in how these cells are playing a role as transducers, as receiving signals and then sending signals to the muscle. And then finally, in terms of just these populations, we did a Surrott clustering at level of 0.2, and we end up with 20 different clusters of monocytes and macrophages from muscle or blood. What's interesting is that most of these cells increase with age in the muscle, except for one population, which is circled here, which is these resident CD206 positive monocytes. They go down with age, and they increase with exercise. So they follow that pattern that we've been seeing all along. And so these are the cells we've been focusing on as maybe key transducers of these mediators of exercise. So I just want to finish with one more kind of piece of data, and that is, so here's a cross-section of muscle, and again, what you're looking at here are these polygonal structures, these large muscle fibers that are responsible for all your movement. Now, one of the well-kept secrets, I mean, it's not a secret, but when we do single-cell RNA-seq, the first thing you do is you get rid of all the muscle. So the cells that are the most dominant part of the tissue are not included in the single-cell analysis. And this is true if you do fat. I mean, so what we're looking at here are rare cells, the endothelial cells, the smooth muscle cells, the muscle stem cells. But all of these fibers, they don't appear in this single-cell RNA-seq data. So we have gone to great lengths to say, okay, we're going to find out what's happening in the muscle fibers, which seems only reasonable. And that's because there's really this increasing field and interest in the idea of muscle as an endocrine organ. We think of muscle, obviously, as a physical tissue for movement, but it's a profound endocrine tissue. It secretes lots of factors, often called myokines, almost all of which have been shown to have beneficial effects to health. So they have beneficial effects on the muscle itself, on fat, on liver, on brain, on tumors, on inflammation. So these myokines are generally thought to be really positive factors. So what we did as part of this big experiment I mentioned, we actually took myofibers, and as shown here on the left, we can actually strip off all these cells, so we're only looking at the myofiber compartment, and then we just did bulk RNA-seq on it. And on the right, you see genes that go up with age or down with age on the left, part of that figure. But one of the genes that was most prominently downregulated in the muscle and then upregulated with exercise is that gene SPP1, which is osteopontin, which is interesting because generally considered kind of more of a pro-inflammatory. But in this case, it looks like it's following that pattern of a pro-regenerative myokine anti-inflammatory. So we looked at this just in terms of protein levels. It definitely goes down with age, and it's more than doubled with exercise in the old tissue. And then here's the one functional piece of data. If we injure muscle, and we show old muscle, and we've shown again that exercise, which is in the middle, enhances muscle regeneration in an old animal. If we use a neutralizing antibody to osteopontin when we're doing the regeneration, it completely blocks the beneficial effects of exercise. So it seems to be a key transducer of what's coming out of the muscle fiber and then mediating this improved regenerative response. So just back to this model of, I think we have to add osteopontin. We're now trying to knock this one gene out in muscle fibers to ask what effect that has on this kind of tissue regeneration and on stem cell function, and really thinking about why exercise is good for us. What really are the cells that are present, what they're secreting, how exercise changes the cell population and the gene expression of those cells that leads to factors that are not only acting locally, but are being secreted into the plasma throughout the body and potentially having these great benefits for health in tissues throughout the body. So just want to, again, stop. So my senior scientist in the lab, Ling Lu, really spearheaded this work on the single-cell RNA-seq study, but a lot of people in the lab have contributed to many aspects of this, and I've mentioned our collaborators. Obviously want to acknowledge the funding sources we've had, and I'll stop and thank you for your attention.
Thanks, Tom. One question. All right, the first hand I saw up was right here. Please speak loudly.
I'll repeat it. Okay.
You know, one of the issues that, you know, the history of sarcopenia in mice and in humans is totally different in terms of the time frame and the fact that in humans there's a strong effect on neurological control, and that's why it's different. Do you expect that this result also apply to humans?
To humans. So yeah, do these effects apply to humans? So mostly what I talked about, the assay we use is a regeneration assay. I didn't actually show sarcopenia because, as you point out, there actually isn't much sarcopenia in a mice over this time frame, so it's difficult to study. But I would say absolutely these myokines and the secreted factors in response to exercise are going, you know, they affect the liver. So I feel certain, I'll go out on a limb, I feel certain that these myokines are responsible for the beneficial effects for preventing or reversing sarcopenia in humans. We just don't know what those factors are. But I have a feeling it's, you know, we've looked at the role of the stem cells in muscle in sarcopenia. I think they don't have much of a role. I think, again, it's going to be the immune cells that are mediating this, and the beneficial effects of exercise, whether it's aerobic or resistance, are going to be mediated by, not directly, but indirectly through the immune cells in the tissue.
All right, thanks, Tom, very
Thank you.
much.